Today we are going to study about agarose gel electrophoresis. Agarose is a linear polysaccharide, average molecular weight of 12,000, and it is made up of repeating unit of agrobiomes. Agarose is a component of agar. Agarose is generally used in a concentration of 1 to 3%. Agarose gels are formed by suspending dry agarose in aqueous buffer and then boiling the solution, then boiling the mixture till a clear solution is formed. This is poured and then allowed to cool at room temperature, and then it forms rigid gels. The gelling properties of agarose are both due to inter and intramolecular hydrogen bonds between long agarose chains. The cross-linked structure gives the gel good anti-convectional properties. Pore size of the gel is controlled by agarose concentration. Low concentration gels form large pore size, and while high concentration gels form higher concentration of agarose is used, then it forms a small pore size of gels. Agarose gels are used for electrophoresis of both proteins and nucleic acid. For proteins, the pore size of 1% gels is large enough so that the proteins can move unhindered, and agarose gels are therefore used in techniques such as immunoelectrophoresis, where proteins are allowed to move unhindered in gel matrix according to their native charge. Large pore gels are used to separate even larger molecules of DNA and RNA, but here the pore size is large enough for DNA or RNA molecules to pass through the gel. But now the pore size and the molecular size of DNA and RNA are comparable, so the frictional effects begin to play a role which helps in separation of different sizes of DNA and RNA molecules. Advantage of using agarose electrophoresis is that agarose has a lower melting temperature, and these gels can be liquefied by heating to 65 degrees Celsius, and DNA samples separated by agarose gel electrophoresis can be cut out of the gel, returned to solution by heating, and then recovered. Generally, horizontal gels are used for DNA and RNA separation. In case of DNA molecules, the charge per per unit length of DNA molecule due to phosphate group is same in all the DNA samples. Therefore, they should move towards the anode because they are negatively charged. They move towards the anode with the same mobility under applied electric field. So separation in agarose gel is achieved due to resistance of the matrix to their movement, which is caused by gel matrix because the pore size is comparable to DNA size. So largest molecules will have most difficulty in moving through gel pores, whereas smallest molecules will move fastest, resulting in separation of different molecules of DNA which have different sizes. While passing through the pores, a DNA molecule will experience a drag. So the longer the molecule, the more it will be retarded by each pore. The gel concentration must be chosen according to the size range of molecules that we want to separate, because different concentrations will form different sizes of pores. Gels containing 0.3% of agarose will separate double-stranded DNA which have a size between 5 to 60 kilobase pair size. DNA are run in horizontal gels totally submerged in buffer solution. Samples are prepared by dissolving them in buffer. Then glycerol is added so that it increases the density, so that the sample solution sinks into the well. And bromophenol dye is added to the sample buffer. It acts as a marker of electrophoretic front. That is why it is known as tracking dye. Sample is loaded into electrophoretic apparatus, that is into the well of the gel. Then electrophoresis is performed. After electrophoresis, DNA is stained by fluorescent dye, that is ethidium bromide. Gel is rinsed in solution of lithium bromide, which is fluorescent, and then it is after washing it is visualized under UV light. DNA band appears as orange-red band. Single-stranded DNA are represented in terms of nucleotide units, NT. Single-stranded DNA shorter than 1000 nucleotides, which differ from each other by 1 nucleotide unit. They can be separated using 3.5% polyacrylamide polyacrylamide gel electrophoresis. 12% gels, polyacrylamide gels are used to separate fragments between 20 to 100 nucleotide units. Electrophoresis of nucleic acid is carried out in submerged gel electrophoresis. Agarose gels are cast in a rig, and when the gel is still in molten form, comb is applied so that it etches the wells in it, and when the gel has solidified, comb is removed and the whole rig is along with the gel is transferred into the electrophoretic apparatus. Buffer is filled till the gel and the electrodes are submerged. Then sample is loaded. When the gel is submerged in buffer, then sample is loaded. Then current is switched on. When the tracking diode, which is the other end of the electrode, because nucleic acid will move from cathode to anode, when the tracking diode, which is the other end, current is switched off and DNA is visualized by staining with lithium bromide. After staining, it is visualized under UV light. Applications is of Agarose gel electrophoresis is determination of molecular weight of the DNA, detection of plasmids. It is used for immunoelectrophoresis to study antigen-antibody reaction. It is used in southern blotting and northern blotting, and restriction mapping of DNA is done by using Agarose gel electrophoresis. Electrophoresis of RNA is carried out in Agarose gel electrophoresis. Separation is based on size. 2% Agarose gel is used to separate ribosomal RNA 18S and 28S. If greater resolution is required, then 2.5 to 5% acrylamide gradient gels are used to resolve transfer RNA 4S and 5S ribosomal RNA. Native RNA can be stained by lithium bromide. If RNA size is to be determined, then RNA is denatured first by heating, and then heat denatured RNA is adducted with formaldehyde or glyoxyl. They form adducts with amino groups of guanine or uracil, and then this DNA and RNA which is denatured, this is stained with RNA which has been denatured is stained with acridine orange.